Welcome to Bio3 lecture again. Today we will talk about nucleic acid. Previously we have already talked about three other biological molecules, or you may call it macromolecules. Okay, the first one we talk about carbohydrate. The second one, lipids. Lipid is a long lecture, remember? And then the third one is the protein. And this is the last one, nucleic acid. These four major types of biological molecules is basically they are the four major types of food we eat every day. And then we eat what we are, meaning that we eat these four major types of food, and then what happens is that we will break down all these polymer, carbohydrate into monosaccharide, lipid into fatty acid and glycerol, and protein into amino acid, nucleic acid into nucleotide, so that our body, the small intestine, can absorb them. After our body absorbs all these monomers, our body will reassemble all these monomers into polymer, so that we use the polymer to build our body. For example, after we absorb amino acid, our body will reassemble, or dehydration. We dehydrate the amino acid to form protein again, so that we can use protein to form our muscle. So that's why I said we eat what we are. And today we will talk about the last one, nucleic acid. Nucleic acid, some example is the famous DNA. DNA is an example of nucleic acid. So we will first talk about structure, function of nucleic acid, and then we'll talk about an example of it. It's called ATP. Nucleic acid is made up of chon again, CHON. That's why they're the four most abundant element. But this time you have a 4P, phosphorus. Nucleic acid, they made of thousands of nucleotides. Nucleotide is the monomers here. Nucleotide is the monomer linked together. We'll show you the pictures here. So component of nucleotide, it has a phosphate group, it has a pentose sugar, and it has nitrogen containing base. Some textbook they just call it nitrogenous base. Nitrogenous base. Nitrogenous base. So what they are, well, let me color code them, then it will be easier for you to see it. Pentose sugar, and then nitrogenous base. So the nitrogenous base is here. The pentose sugar is this one here. Last but not least, the phosphate group is here. Phosphate group is negatively charged. This is very, very important. We see the negative charge here at one of the oxygen, actually it's this one here. This one here, sorry about that, it is not this one here. This is connected to the carbon. And this is another negative charge. So usually the phosphate group is 2-. It contains 2 negative charge. One negative charge on one oxygen atom, the other negative charge is on the other oxygen atom. That's why it is usually 2-. It is important to know that phosphate group is negatively charged? Because it makes the DNA molecule negatively charged. Yes, DNA molecule is negatively charged. This is something important for you to know. Okay, let me write it down. DNA is negatively charged. Okay. Pentose sugar. Why do we call it pentose sugar? Because it is in the form, it has 5 carbon. Or it is in the form of pentagon. Pentagonal shape. And it has 5 carbon. But the 5th carbon is here. The 5th carbon does not make the pentagon shape. It is the oxygen atom make the is part of the pentagon shape. The other 4 carbon, they're connected here. So remember that the 5th carbon, the 5' carbon, connects to the phosphate group. Okay. And then the nitrogenous base, the nitrogenous base connects to the primary, the 1'. We call it 5' carbon connects to the phosphate group. The 1' carbon connects to the nitrogenous base. We'll talk more about nitrogenous base in the next few slides. Don't worry about it. Okay, these are the 4 types of nucleotides. What do you mean 4 types? Actually, these are the 4th type. 5 types of I don't want to say nucleotides. I would say 5 types of nitrogenous base. Nitrogenous bases. You have adenine, guanine, cytosine, thymine, and uracil. This is the tricky part. Adenine always form hydrogen bond with thymine. Guanine always form hydrogen bond with cytosine. Adenine and guanine, we call them purine. Cytosine and thymine, we call them pyrimidine. Pyrimidine. So for DNA, we have let's see. For DNA, we have for DNA it uses these 4. The first 4 nitrogenous base. For RNA, it uses A, G, C, not T, but RNA uses uracil. RNA uses uracil to replace thymine. So let me write here. RNA uses A, U, because they form the bond together, and then GC. Oh, well, let's do it this way. This is the way I usually do it. G, C. And what about DNA? DNA A, T, G, C. Why do I put it this way? Because A form hydrogen bond with T, and G form hydrogen bond with C. And then A and G, they are purine. Okay, I'll just put Pu here. Pu, purine. And then T and C, they are pyrimidine. I'll put Py here. So how do you memorize the base pairing rule? Very simple. Remember in West LA College, we have GC building. GC building stands for General Classroom, right? General Classroom. So in West LA College, we have, so you can call it at GC building or at General Classroom building, at GC buildings. That helps you to memorize. Or if you are a professor, professor level, we usually memorize it instead of at GC building or at General Classroom, we call it at Gentleman's Club. So this is how we memorize it at Gentleman's Club. Okay, these are the structure of adenine, guanine, uracil, thymine, and cytosine. As you can see that adenine and guanine, they have a similar structure. That's why they belong to purine. Let's draw a line here. These are called purine. These type of molecule we call purine, which is made up of one hexagon ring attached to a one pentagon ring. And these uracil, thymine, and cytosine, they are called pyrimidine. Pyrimidine. So they are a hexagonal ring. As you can pay attention to uracil and thymine, remember thymine, you only see it in DNA. Uracil, you can see it only in RNA. Oops. Spell it wrong, sorry. RNA. So what happened is that they have very similar structure. The only difference is this part here. Thymine has a CH3 group attached to this carbon, but uracil, we omit it. We scientists are very lazy. It has a hydrogen atom attached here. You are too lazy to write out the hydrogen atom, so this is the differences. Thymine has a CH3 group. The uracil has a hydrogen atom. Oops. Okay, they already put it here, so I don't need to write out purine, pyrimidine, and CH3. Actually, it's on the slides. Okay, all right. Well, but I will still put it here. This is a hydrogen atom, and you have a red circle here. Okay. So that's the differences. So base pairing rule are